So we're happy to welcome Fela Bouchama, who is also a first-year PhD student. She will be speaking to us today about understanding how coacervation of two viral proteins drives the formation of membrane-less compartments. So please begin, Fela.
Thank you, Henry. Hi, everyone. Today I am going to introduce you my subject, as he already said, about understanding how coacervation of two viral proteins drive the formation of membraneless compartments. A quick overview about my studies. I had a baccalaureate degree in science in Algeria, where I did my high school studies. After that, I've got a bachelor degree in biochemistry and a master degree in drug science in structural biology field in Montpellier, in France. I did my Master 1 internship at the Thorax Institute in Nantes about protein remodeling and structural bioinformatics. And I did my Master 2 internship at the ILL on the same subject as my PhD. My project is a collaboration between the large-scale structure group and diffraction group from the ILL and the viral replication machine group of the IBS. I am supervised by Sylvain Prévost and Gabriel Coelho at the ILL and by Marc Jamin at the IBS. I will start by introducing you the subject and background, then the objectives, the results, and the next step to undertake. So viruses are obligate intracellular parasites of a very small size. They are too small to be seen by light microscopy. They constitute a very elaborate form of parasitism. They can reproduce within living cells. Having no energy system, they divert the cellular machinery to their profit to replicate and ensure their survival. A virus particle consists of nucleic acid made from DNA or RNA, so we can call them a DNA virus or an RNA virus. This genome is surrounded by a protective coat of protein, and the vast majority of viruses have RNA genomes. They need their own RNA polymerase to copy their genome, as there is no equivalent enzyme in the host. Being totally dependent on a host cell in order to survive, viruses have a common cycle which starts by the entry of the virus inside the cell. The interaction between protein on the surface of the virus with protein on the cell is the first step of the cycle. The genetic content of the virus particle is then released into the host cell, where replication of the viral genome may commence. A virus must take control of the host cell replication mechanism, so the messenger RNA produced is used to instruct the host cell to make virus proteins. At the next stage, newly synthesized genome and protein are assembled to form new virus particle called virin. Finally, these new virus particles are released from the cell. Different viruses infect different cell types, tissue, or organs, causing a wide variety of diseases with different degree of severity. These diseases can be treated by antiviral drugs or vaccines. However, some viruses are capable of both avoiding the immune response and mutating to become resistant to antiviral drugs. Zoonotic viruses that emerge from wildlife and domesticated animals pose a serious threat to human and animal health. Over the past century, humanity has witnessed the emergence of infection that have resulted in varying numbers of human fatalities. As you can see in blue, influenza viruses that originate from birds account for an important proportion of these deaths. And recently, many zoonotic viruses that originate in bats, such as Hendra virus or Nipah virus, and the reason why I am behind the screen right now, the coronavirus, have caused outbreaks with high mortality rates. The viral genome can vary depending on the virus. Human cells have double-stranded DNA and replicate via single-stranded RNA. Viruses, however, are not so simple, as they can be either DNA or RNA viruses. But each can also be single or double-stranded. The structure of a virus's genome determines how it will replicate, and this is the basis of the Baltimore classification. The figure below is the basis of this classification. It is first divided into, depending on how the virus replicates with or without the reverse transcriptase. Then for viruses that replicate without reverse transcriptase, further segregation is made depending on whether the genome is made of DNA or RNA, and then whether it's double-stranded or single-stranded. And this is how we have 7 different classes of viruses, and the replication of the virus within each group is supposed to be similar. Many current viruses come from different viral classes according to the Baltimore classification. The coronavirus and the hepatitis A, which come respectively from the coronaviridae and picornaviridae families, are both from the class 4. My PhD research is focused on a virus from the class 5, which is the rabies virus. The rabies virus is a neurotropic virus which enters the human tissue from the saliva of a biting animal and causes fatal encephalomyelitis. And despite the existence of an efficient vaccine for more than a century, it continued to kill more than 50,000 people every year. Early diagnosis is difficult and it's always fatal when it's reported. The risk of rabies is highest in countries including most Asia, Africa, and Latin America. The viral particle of the rabies virus is composed of 5 structural proteins. The G protein that binds to the cells, the matrix protein, which is located on the inner virus envelope, and we can find inside the nucleoprotein here, which binds to the RNA tightly with the polymerase L and its cofactor, the phosphoprotein. As you can see here, the polymerase L works only if the nucleoprotein in green is encapsulated by the RNA. The viral cycle of the rabies virus is cytoplasmic and common to other viruses cycle. After the receptor binding on the membrane, the virus enters the cell via endocytic pathway. The viral envelope merged with cellular membrane here. And after that comes the replication phase where the virus makes copies of itself, and then the transcription of the RNA messenger corresponding to the 5 viral proteins. It was recently shown that the phase of transcription and replication of rabies virus take place in cytoplasmic inclusion that we called Negri bodies. The final step of the cycle is the release of the viral components from the cell. In 1994, Negri described the presence of cytoplasmic inclusion in cells of people who have died from the rabies virus. Nowadays, we can check if someone died from the rabies virus by visualizing these Negri bodies in the neuronal cell. These bodies are the sites where viral transcription and replication of rabies take place. We call them viral factories. The images below shows a time analysis of a cell infected by rabies virus. We can see that after 16 hours of infection, Negri bodies in green grow during the viral cycle around the nucleus in blue, before appearing as a small structure at late stage of infection. These Negri bodies are membraneless compartments and have properties of liquid-liquid droplets since they are formed by liquid-liquid phase separation. Similar inclusions are found in cells infected by related viruses like the measles virus or the Ebola virus. The Blackledge group at the IBS succeeded in forming droplets by phase separation in vitro by mixing the purified protein of measles virus, which forms the Negri bodies. Cells and cellular organelles are typically surrounded by a lipidic biomembrane, which controls the exchange of molecules between the separated compartments. However, some subcellular compartments doesn't have a surrounding membrane, but still perform specific biochemical functions, like the nucleus or the centrosome. When it comes to why and how the cell creates and uses these membraneless organelles, there are still more questions than answers. Recent studies have shown that membraneless compartments have properties of liquid and are formed by liquid-liquid phase separation. And despite the absence of a membrane, these organelles can be very large in size and have a multi-layered organization with highly dynamic structures. In addition to the primary polymer that make up the membraneless organelle, small molecules, protein, and nucleic acids can potentially enter the structure they will be absorbed or excluded depending how it will interact with the environment. The liquid-liquid phase separation is a process which results in the coexistence of two different phases: a condensed phase in which the macromolecule are concentrated, and a second one in which they are diluted. There are no visible separation between these two phases. And it can be induced by multivalent interaction between protein composed of modular interaction domain or intrinsically disordered region, or by RNA or DNA molecules. However, little is known about the organization of the condensed phase or the physicochemical properties of it. Simply, a solution of charged macromolecules which undergoes liquid-liquid phase separation is known as conservation. A recent studies shown that the co-expression of the nucleoprotein and the phosphoprotein of the rabies virus in vitro lead to the formation of cytoplasmic droplets and can reconstitute the liquid-liquid phase separation. So the aim of our project is to try to reproduce this phase separation by mixing purified nucleoprotein and phosphoprotein in the first step, and in the second one, to use neutron scattering and diffraction experiment to probe liquid-liquid phase molecular process at different time and length scale. So to do that, we need to produce both purified phosphoprotein and purified nucleoprotein. And finally, we need to reconstitute the nRNA complex. For the nucleoprotein that we call N0P68, we use a complex between the nucleoprotein in green and a peptide of 68 amino acid tribe for the N terminal of the phosphoprotein. So we started by the expression of the phosphoprotein using a bacterial expression system, which consists on the use of DNA coding for our phosphoprotein. And this DNA is inserted in the plasmid expression vector, which is then transformed into a bacterial cell. And then the cell propagates and produces our phosphoprotein, which can be purified next. So for the purification step, we used an affinity column, which is a biochemical method of separating based on the highly specific interaction between a histag that we inserted in our phosphoprotein and the methyl ion. It can be used to purify and concentrate a substance to reduce the amount of unwanted substance in the mixture. We also used another method of purification, which is the size exclusion chromatography, to separate the protein in solution by their size. The final step of purification is the use of an SDS gel, which is a denaturing technique that separates protein according to their molecular weight. After the separation, the visualization can be carried by the coma C blue colorant. So here you can see a fraction of our protein before the purification. We can see the appearance of a big band with a presence of an unwanted substances. And here is a fraction of the protein after the purification, where we can see the appearance of only one band corresponding to the molecular weight of our phosphoprotein. So now comes the expression of the nucleoprotein N0P68, and for that we use the same bacterial expression system as for the phosphoprotein. And after purification, we can see the appearance of a band above, which corresponds to the molecular weight of the nucleoprotein. But you can notice that the band is not as thick as the one that we had for the phosphoprotein, which means that we have less amount of proteins. So we tried another expression system different from bacteria, which is insect cell system, where we infect insect cell by recombinant virus coding for the nucleoprotein. And you can see that after the purification, we obtain a thicker band attesting for bigger amounts of proteins. Well, when N0P68 is expressed with synthetic RNA, the part corresponding to the 68 amino acid leave the complex as we break the bond of these complex. And the nucleoprotein polymerizes into the RNA and form either circular complexes called rings or long ribbon called nucleocapsids. So the presence of the nucleocapsids and rings was verified by negative stained electronic microscopy. Here, the first image shows the N0p68 without RNA, and we can see that once we add the RNA, a small RNA, we see the appearance of rings, and with a bigger RNA, we can see the appearance of nucleocapsids. So now that we have been able to produce purified phosphoprotein and purified nucleoprotein complexes with RNA, we can try to visualize the phase separation in vitro by mixing both protein inside tubes. And the transition to turbid solution inside these tubes would attest phase separation. The visualization of the liquid droplets can be done with light microscopy. So the next step would be to vary the condition for the phase separation assay and to try to have a better understanding of the state of the various components in the dense phase using neutron scattering experiments. And since little is known about the organization of the phase which compose the liquid-liquid separation, small angle scattering would help us identify the morphology of protein, whether we have complexes or not. And since we have a complex system containing two different proteins, we may use construct variation to have information about each protein and the complex organization. Thank you for your attention.
Well, thank you, Fela, for that nice presentation. Now we will proceed to questions.
Hi.
Yeah, so I have several questions. First of all, why do you think that you need the nucleocapsids assembled for droplet formation and you never tried to do it with the N0p, with phosphoprotein?
Okay. Well, actually, we are not sure. So we are going to do the different assays with the N without the RNA and with the N complexed with RNA. So we actually don't have the real information if it's going to work only with RNA or without RNA. And
okay.
The question was?
And then, could you explain actually which RNAs you used for those negative stain pictures you obtained? Like the short one and the long one, and what was the sequence?
Actually, the long one was a poly-A. We didn't know exactly how much A was in the RNA because it was a long time ago inside the fridge. So we tried just for a first time to be sure that it can reconstitute either rings or nucleocapsids. And for the small one, if I remember, yeah, it was an RNA from VSV that was used already from another PhD student before me. And now we are ordering the new RNAs with specific sequences.
Okay, thank you. And the last question is about the phosphoprotein. So on the gel, you actually had like a double band. So did you tested it? Why do you have a double band? What it is related to?
Which one was it? The phosphoprotein?
Phosphoprotein, yes.
Yeah, actually, it was just a gel problem that we had.
Okay.
Like this one because it was showing the bigger band actually.
Okay, thank you.
You're welcome.
Hello?
Christian.
Yes, okay. So I am a postdoc at the ILL. I have one question concerning the contrast matching for the SANS with the LLPS.
Mm-hmm.
Have you already, well, as far as I understood, you have not yet conditions where you have the LLPS?
No, we are in a step where we produce protein, and that would be the next step. Once we have the droplet, we will try to deter the protein. So we did not do that yet.
Well, okay, but the plot here is that you are showing, well,
no,
that you're
this is
changing
just a representation of, in case we have.
Yes. No. Okay, well, what you should keep in mind that you might change the system by changing the solvent. So from D2O to from H2O to D2O, this might strongly influence your system, and this might actually kill your LLPS conditions.
Yeah, we will talk about it.
Which might then lead to problems with the contrast matching.
Okay, thank you. There is another person who has raised her hand, Francesca, but I'm not sure if it's a question or a comment. Are you there, Francesca?
No, it's a question. I'm definitely curious about the production of the protein using insect, and particularly, I think if I understand well, is the protein that you intend to deuterate. How?
Yeah, I know it's impossible. We don't know if we can do that inside insect cells, but
Okay, because I was really curious if there was already a protocol about
no,
forcing
actually,
an insect to live in deuterium.
we speak a little bit about it, but I don't really know if it's going to work. So if it doesn't work, we can try to have more protein with the bacterial expression system. It will be kind
Okay, and this is really another question, but in your opinion, virus is living things or is not?
You know, my boss told me that someone is going to ask this question. Well, in my opinion, I don't think so, since they cannot reproduce by their own. They need a host cell to survive, so I would say no.
Okay, it was my questions.
So what's your opinion? Do you think it's a living?
I have no clue. I don't know, because other parasites cannot reproduce themselves without an host, like wasp. So I don't have a clear mind.
Actually, it's a big. No one is okay with the other, so I think so.
So I'm neutral. I have no clue. Okay, this is everything.
Thank you, Francesca. I think it's probably a matter of opinion. So my little question is, I'm not in this field, but my question is, if I have the history right, Pasteur developed the rabies vaccine relatively quickly and simply, at least as compared to what we're hearing now in the efforts of developing a coronavirus vaccine. So is it because the rabies vaccine was in a particular class that made this easier? Or was it not so easy as I've had the impression? But he had more primitive techniques, and so it does seem like he was able to develop and test a rabies vaccine sooner than what we're doing now with all the scientists in the world working on the coronavirus. Can you explain?
Well, I would say that how the rabies virus works is easier to interpret than the coronavirus. But if it was difficult or easy for him to find, honestly, I don't know. But
okay, I saw a documentary. I should look this up on the web, I suppose. I saw a documentary which I think was factual enough that said that he used some dried out infected tissue and then maybe heated a little bit to assure that the virus was killed. Again, it's a matter of a definition if it had ever been alive. And then put it into solution. And I think he used that basically as the vaccine. So perhaps he was taking a lot of risk. But do you happen to know how he developed it?
he actually used, I think he used rabbit tissues from what I know. And he did, how to explain? And these tissues are supposed to awaken immune response into the human body. So I think it seems simple, but I don't know if it's that simple, actually.
That's what I thought he had done. So apparently the same simple technique cannot be used to produce a COVID-19 vaccine.
No, he actually did the simplest one. And after that, with technology, they perform I don't know where they did exactly, but it's better now with better results.
Okay. Okay. Yeah, thank you. Okay, thanks again to our speakers and to all of you who have participated. And we'll meet again in 1 week's time. Have a good evening.
Have a nice day.